We've got David, who's going to tell us all about genetics and everything.
Thank you very much, Chris. So I love coming to Cold Spring Harbor. That's the first thing. I looked on my computer as I was putting this talk together and saw that I did the first talk in this course about a decade ago. And also there have been many other meetings that I've been able to be at in this room. And this is just the most awesome thing as the contractor that Anto and I likes to say. Because you know, here you are in a place where Jim Watson hears the talk and says, so that everybody can hear, I don't believe this. And here you are in the place where the genomicist, Kari Stephenson, goes toe-to-toe. And this is the place where I met Svante Pabo, you know, this guy who charged out into the freezing cove when the water was really cold, not like it was last night in his swimming. This is really an amazing thing. And I thought that last year it took an interesting detour because there we were in Barcelona. And so it was, you know, our summer camp, but, you know, with quite a difference. And now we're back. And we're back. And unfortunately, I'll be gone after this afternoon, but usually on the last day, Chris issues bows, and there's a contest. And so you can look forward to that. So I actually have a massive responsibility here, which is that looking at the program, I need to represent, as we like to say, genetics and genomics. And so that means that in addition to some of the state-of-the-art, I want to make sure that we have a chance to discuss foundations and any general issues, including some of the topics that are up here on the board, like what to do about very large-scale data, and you know how you might be thinking about taking part in big sorts of genomic collaborations, because they're all huge. And also, if you are in these, what about harmonizing phenotypes across data sets? All of these issues are intrinsic to modern-day genetics and genomics. These were the major issues that I was hoping that we could cover in this talk. Why it is that people do vary in their liability to addiction, or why they should be liable at all to addiction. What is the environmentality of addiction, the heritability of addiction, the types of genetic variants or alleles, rare, common, regulatory coding that are predisposing, where we are in addictions genetics, what it means to do genetics and genomics as opposed to doing molecular biology and any molecular neuroscience, which all of you are going to be invested in, probably. The dependence of allele effects on context, the new sort of linkage of genetics to the neuroscience with increasing use of neuroscience-derived phenotypes instead of these very crude clinical phenotypes or broad phenotypes like happiness, which you can do linkage on. I'm actually being interviewed by a reporter tomorrow about a paper that I'm mysteriously involved in where we did a genetic linkage to happiness. I wish we hadn't, but I'd be happier if we hadn't, but you know, there you are. The contexts also include population isolates, and they very much include the model organisms. And I'm going to be concluding with a use of next-gen sequencing technology and in the deep sequencing context in humans and in a model organism, namely the rat. That's if we get through all this. So first of all, we need to start with heritability, because this is the broad justification for doing genetics and genomics of addiction. And of course, this is the famous Adams cartoon, which was in the New Yorker. And it shows these two twins who look remarkably like the famous twin geneticist, Tom Bouchard. It shows these twins with the same cockamamie invention, and here they are. They've met for the first time in the patent office and very similarly dressed. And you might think that behavior can't be so powerfully genetically encoded, but in fact, as quoted in Bouchard's book, there are a number of instances in which twins separated at birth met decades into their lives and discovered that they had had very similar life histories. They had both become firemen, they had both married women named Kathy, they looked about the same. So, you know, really quite amazing convergences of behavior based upon genotype. And anyone who has experience with certain breeds of dogs or cats that have highly distinct behavioral differences, very complex behaviors, you know, things like tunnel digging, things like pointing, very complex behaviors can be not only strongly genetically influenced, but also almost encoded. And it happens to the largest extent when people face an average sort of environment or can at least choose their exposures so that genotype becomes more paramount. So here are these disorders that depend upon the choice to take to use an addictive agent and also the availability of the agent. And so obviously they're environmental disorders, and yet they're moderately to highly heritable. This is a meta-analysis of epidemiologically conducted twin studies for a series of addictive disorders, including gambling. So it doesn't have to be a drug. And in each case, the heritability, which is the fraction of the total variance in liability that is attributable to additive genetic factors, the heritability ranges from about 40% to about 60%. So about half of the inter-individual variation in liability to addiction is due to genic causes. And the remainder isn't just environment. A lot of the remainder is measurement error. A lot of the remainder is interaction between genes and environment. So this is an enormous influence of genetic predisposition on outcome for what we would superficially think are going to be environmental and choice-determined disorders. There are other interesting things that one can observe here. First of all, there are many thousands of twin pairs that are included in each one of these categories. So these composite estimates are probably about as accurate as we're going to readily get, but also there's enormous variation across studies, as shown by these ranges here. So from one study to the next, you might get a very different heritability value. The other thing to observe is that instead of it being the least addictive substances or agents for which the heritability is highest, that it tends to be the other way around, that the substances and agents that are more likely to, let's say in an animal model, elicit punishment-resistant responding, those agents have the higher heritabilities. So this is pretty much what's known about heritability as far as most people are concerned, that there is heritability, and oftentimes it just kind of stops there. But then we have to think about why there is this heritability. Well, there's heritability because there is DNA variation that is shared more in common between people who are at closer degrees of genetic relationship. So what's this variation? Humans have have over 22 million common genetic variants. And eventually every nucleotide in the genome will probably be found to be genetically variant in someone. So if somebody asks how many single nucleotide variants are there, you could make a reasonable guess that there might be at least 3 billion, since that's our number of nucleotides. But within each one of us, there's a certain level of genetic variation because we have different chromosomes inherited from our mother and our father, and on average, we each have about 3 million heterozygous chromosomal sites. That is a nucleotide at which we have one allelic form from our mother and the other allelic form from our father. And so this average heterozygosity, this 1% average, 0.1% average heterozygosity overall is actually a pretty good estimate of the overall variability of humans as compared to other species, and in fact as compared to wild mice, wild salamanders, lots of other species. We're not that genetically diverse a species, but with this enormous amount of genetic variation, we're nevertheless all neurogenetically individual. And furthermore, most of the human variation is between people rather than between population groups. Quantitatively, only about 10% of genetic variation in humans is accounted for by looking at variation across populations, rather than variation just from one human being to the next. Some of these variants are highly significant. There are hundreds of large copy number variants in each of us, and also each one of us carries on average over 10 stop codons, which are equivalent to gene knockouts. And in fact, this is going to be the stop codon roadmap of this talk. It's not the real roadmap of this talk, but during the course of this talk, I'm going to talk about 2 stop codons which have been discovered to alter impulsivity in people, and also a GRM2 stop codon on the metabotropic glutamate receptor 2 that we found is common in the Wistar rat and is selected in some of the rat addiction model. So how do these genetic variants work to alter behavior? First of all, are the variants that alter behavior rare or are they common? And we'll discuss that. But right now, do they act alone or do they act in combination? Everything that we hear at this meeting would encourage us to think that they act in combination. And that's because the brain is such a complicated system, and to make a cell requires the action of thousands of genes, and for these cells to interact together in the ways that they do requires many different molecular players. So you would think that for a behavioral variant to alter a behavior, that it would need to be cooked up in the right combination of ingredients with some other genetic variants, and then the cake would be baked. And so this is how you would think that that would all work. And this has been a very seductive idea, the idea that that there is epistasis, that the variants are conferring risk only in combination. And this is very much like the concept that was explained in the first Batman movie, where the Joker terrorized the citizens of Gotham City by cooking up these chemicals which were put in cosmetics. And if you only used the poisonous lipstick, no problem. If you only used the perfume, no problem. But in combination, several of these things would result in you dying with this terrible grin on your face. And so these were the Joker products that were used to terrorize the citizens of Gotham City. Now the other model is the genetic heterogeneity model, that different variants are conferring risk in different people. And this model could also be called the CAR model. you know, you go out to start up your car in the morning and you turn the key and it won't start. And so, you know, if you're a certain type of person, you have to open up the hood as if you could figure out what's going on in there. And you look in there and there are all these complicated parts. And you realize that what has happened is that it's insanely complicated, but the thing didn't start probably because one thing was broken. And you don't know what that thing is, since this is the heterogeneity model, that many things could go wrong, but probably there's one key thing that went on. So here let's start to look at the ability of genetics to make not only correlational observations, but causal inferences. And this is a big strength of genetics, that you can, if you do the analysis correctly, make an inference about causation and about also the mechanism of causation. So here are the two models then. Here's the epistatic model. People are affected with an addiction because they have one of a number of different combinations of bad alleles or addiction alleles. And here's the heterogeneity model. There are again many puzzle pieces, but one is sufficient to cause the problem, any one of those. And so they're all different flavors, but ultimately they're of single origins. So genetic analysis can distinguish between these two models because we can look at how similar are people who are at different degrees of genetic relationship. And if, for example, they are monozygotic twins, no matter how many alleles are in the combination, the two twins will share that full combination. So there could be 100 of them, and they'll share 100 out of 100. Here in the monozygotic twins, and under the heterogeneity model, again, there's only one locus involved, and again, the monozygotic twins will share 1 for 1. But as soon as you move to the dizygotic twins, the story changes, because if there should be 4 required, the odds of the second twin receiving that combination, that exact combination of 4, would be 1/2 to the 4th power. And if you had a higher order combination, then the odds would be even less. Now, of course, there might be multiple combinations that are possible, but nevertheless, you would expect very different proportions of identity in monozygotic twin versus dizygotic twin under the epistatic model than under the genetic heterogeneity model. Under the genetic heterogeneity model, if it's a dominantly acting allele, you expect a 2-to-1 ratio. If it's a recessively active allele, you would expect a 4-to-1 ratio. It just, you know, directly follows from what it takes to get sharing in monozygotic twins and dizygotic twins. And you can also look at individuals who had other degrees of a relationship. So now we take these. Yes, Jonathan.
What happens if it's both? I mean, the assumption you're making a clear, it's either or, but it could be both.
Then it's an issue of proportion. If it's both, then it's an issue of to what extent that the single allele effects dominate versus the multi-allele epistatic effects dominate. So that would be my answer to you. So you have to go to this next step and look at the empirical data and ask how closely do the data fit one model or another. So for example, if you go to autism, the MZ to DZ ratios are 100-to-1. So the ratios do not fit the single locus model. They fit a multi-locus epistatic type model quite well. You can't explain those data with a single locus model. It's either new, or at least unless there's new mutations, which people do believe that there may be. But it doesn't fit that model well. It fits much better the epistatic model. But if you have, as you see here, for the addictions, close to 2-to-1 MZ to DZ ratios, and you don't have an 80-to-1 or 50-to-1 MZ DZ ratio, then it is difficult to argue that a large proportion of the inheritance of addiction is non-additive. And so this is why, and this is actually throughout psychiatry. This isn't just addictions. Elliot Gershon will tell you that there's very little evidence for epistasis in bipolar or schizophrenia. Also, people are always talking about it, but the actual data don't seem to bear it out.
Well, but if you look at flies in terms of response to alcohol, you know, I'm just talking about treating McKay's work.
Yes.
She will say that there's a large component that's epistatic, at least in terms of response to alcohol. I'm not saying addiction, I mean, and the response is not addiction, but it's other responses to alcohol.
More needs to be done. These are the data that we have available today. At this point, we're stuck with these 2-to-1 MZ to DZ ratios. And the argument that I would make is that in advance of discovering the functional loci and then studying them alone and in combination and measuring their additivity or non-additivity, that it is wrong to assert that the inheritance of addictions is an epistatic inheritance.
But I'll give you an example in your own data, right? I think it was the knockout for the MAO or the
Yes.
5-HT1B, right? And initially it looked like, I think it was in 129, right? And then as it was back crossed into a different background, if I remember correctly, it disappeared. So it, and I'm saying this is anecdotal because I don't remember the expression.
No, you're absolutely right. And there have been a number of single gene effects that have been shown to disappear on other genetic backgrounds. This is a measure of how it works across the broad population of addicted individuals who were studied in places like Virginia and Australia, but certainly within any of these addictions, you would expect to find that there would be interactions of that sort that has actually been seen with some of the knockouts, which work differently on different backgrounds.
Right. So just making the point that,
Yes.
that there is some type of gene-gene interaction here, and that this type of analysis is not revealing it.
Yeah, the strength of this analysis is that this is the overall this captures the overall quantitative effect of epistasis, or as some people call it, polygenicity, versus single locus additive effects. That's the strength of this. And so if you're then going to be looking for the interactions, what it might be telling you is that it might be hard to do that. It might be hard to find those. You might have to be quite clever about it. So what about the predisposition? Is it substance specific or is it a general predisposition? That can also be measured in twin studies. And the way that that's done is by looking at cross-inheritance in twins. You find that there's there's one twin who has nicotine dependence, and ask, does it make it more likely that the co-twin is an alcoholic? And so what you're doing is that you're looking at the inheritance of one thing predicted by another, cross-inheritance. And so similarly, there is cross-inheritance of breast cancer and ovarian cancer due to BRCA1. And the same gene, and it causes cancer in two completely different organs. Is there cross-inheritance of addictions? And it turns out that there's substantial cross-inheritance of addictions. I'm not going to show all of that data, but just let me tell you that deeper and below the simplifications that I'm describing here, there's a big depth of knowledge. There are 3 large twin studies: the Vietnam veteran study, the World War II veteran study, and the Virginia twin study, which all show profound cross inheritance of addictions, especially between nicotine and alcohol.
So David,
Yes.
Marina brought up the
Mhm,
point
mhm.
of interuterine nicotine being a factor, which, so how does that interact with your MZ, DZ?
Yeah, there are all kinds of, actually there are all kinds of
Caveats?
factors like that that play havoc with the MZ.
Well, maybe that explains some.
DZ studies, which could, because, you know, so for example, there's assortative mating between people with addictions. And so right there, one has violated some of the assumptions of twin studies. And furthermore, the two twins will have received the same intrauterine exposure. Over the years, there have been wars fought as to whether that the heritabilities of behaviors are due to common exposures of that sort. But let me just point out this one thing, that the two MZ twins share the uterus, and the two DZ twins share the uterus, both. But the DZ twin concordances are usually about half for behavioral traits as compared to the MZ twins. And furthermore, one can get at this a little bit by looking at the concordances between siblings who are also 50% identical but weren't actually in the uterus at the same time, although they do have the same mother who had the same habits. And very often the concordances between siblings are very similar to the fraternal twin concordances. But we're not out of the woods on
No,
caveats like
I was
that.
just thinking it might mask some of the epigenetics.
It would.
Yeah.
It would.
That was my-
So I'm just going to summarize here this bottom line, that in addition to specific risks which have been found, and I'll show you a genetic factor in alcoholism that confers a substance-specific risk, that there is a general addictive liability conferred by some genetic variants, and both are out there to be worried about. So now, why should some people in particular be liable to addiction? A evolutionary biologist, Theodosius Dobzhansky, said nothing in biology makes sense except in the context of evolution. What would you expect him to say? But it's absolutely true that unless you look at the evolutionary perspective, that you can't understand human addiction. Seemingly, addiction makes perfect sense that it would occur because there are these circuits in the brain that could be hijacked. But really, why would the brain want to tolerate that? It can't be good to be addicted. So why should why should that exist, and why should human brains then sometimes be differentially vulnerable to addiction? Well, addictions are ancient. It isn't a new phenomenon. Here's the drunkenness of Noah, depicted in Michelangelo's Sistine Chapel fresco. So people have always recognized the problem. And here are the explanations. It's not a peculiarly human characteristic. Other species display the differential vulnerabilities to punishment-resistant reward responding. Unfortunately, although we have very good model organism models, none of them are exactly equivalent to the human addiction diagnoses, which have just been revamped in DSM-5. And that's because the human addiction diagnoses have requirements which really you cannot exactly capture in an animal model. But they're very good animal models of addiction, I believe. Many believe that because they're plants and animals that bite, sting, and poison us, that we've evolved resistances to these substances, and that those have made some brains different than others. But also, liability is probably an emergent effect of variation that is maintained for other reasons. So, for example, some people are predisposed to be more resilient to stress, others more vulnerable, others more sensation-seeking, others less. And this is the idea that there are counterbalancing advantages to many of the genetic variants, particularly if it's a common functional genetic variant that alters behavior. A genetic variant such as ones that I'll be discussing, like COMT or the serotonin transporter variant, others that have allele frequencies of 20, 30%, so that a third of the people in this room would be one genotype, a third another, and almost a third another. Those sorts of variants are maintained by balancing selection and for these counterbalancing advantages in behavior. Frequency-dependent selection, niche-specific selection, time-specific selection, as populations are exposed to different forces. And then there's the rare variant category. And this is the category of variants, the severe variant, the genetic glitches, and it's increasingly recognized with the ability to deep sequence hundreds and thousands of people, that there are enormous numbers of these genetic glitches, these rare and uncommon variants, which are predisposing. So now I'm going to move on to the environmentality of vulnerability to give it equal time, if you will. It's important to recognize that usually environmentality of vulnerability is not directly measured. The heritability is measured in the twin studies, but you can't subtract that from the total variance and be left with environmentality. That also includes measurement variance in these interactions. We know when it has been directly measured. For example, in adoptive studies where you adopt a child away from the biological family where there's a biological risk to a foster family, we know that there's a minor role of shared family environment in addictions, which is quite surprising, but true. This is going back to the adoptive studies of Michael Bowman and also Donald Goodwin. But there are many family factors that are crucial, and the breakthroughs in prevention could emerge from recognizing factors that are quantitatively relatively minor in a population sense, but are very important if they would be applied in a deliberate fashion. There also are major interpopulation differences observed for all addictions, and there's a major role for stress and trauma. Stress and trauma is a factor that actually increases the risk of most psychiatric diseases in a fairly dramatic fashion, especially if it's early life stress trauma. So here is the transnational variation in an addiction. This is alcoholism, and the surrogate phenotype is cirrhosis deaths, which are also due to viral infections of the liver, but for the countries represented here, alcohol is the major factor. And then on the x-axis, the per capita alcohol consumption liters per year. And you can see that it's pretty much alcohol in, cirrhosis out. There is a relationship with a correlation coefficient of 0.5 or so. So it's a pretty good correlation between the availability of addictive agents and their use. And by and large, about half of agent use is by people who are addicted. And so, today America is conducting new experiments where we're legalizing marijuana. And by this sort of model, the prediction is that if we plotted some sort of marijuana-related phenotype on this axis, whatever you think about it, that will move up on the y-axis the consequences of that. And if we made laws more draconian or educated people or whatever to discourage use, then you can push it down. So here for alcohol and alcoholic cirrhosis or cirrhosis, the U.S. is just about in the middle of the pack, and it is a manipulable phenomenon. The manipulation was done twice in this country with 2 amendments to the Constitution. There was a temperance movement. It was very powerful, and culminating in Prohibition in 1919, there was a dramatic drop in cirrhosis deaths. And then after World War II and the veterans returning, cirrhosis deaths popped back up to their former level. And there are many types of epidemiology studies that show the availability of outlets and price and convenience all affect use. Yes?
Just one question about that
Mm-hmm.
figure. You show prohibition and there's almost an immediate effect, right?
Ah, but I was careful because I mentioned that the temperance movement, even prior to 1919, it already had a big effect because they were all the women were already marching. They were protesting outside of saloons. They were creating a different environment for alcohol consumption. That's the argument. But I agree with you that it seems like it's still too quick.
Right.
It seems a little too quick.
That was my.
Yeah, it does seem a little too quick, but there was a big antecedent to this. I had the slide. It has a lot of pictures of Carrie Nation and people like her, but I didn't put it up here.
You have a dramatic drop in 5 years.
If this was the only scrap of information that there's temporal variation in addiction, I think it would not be worth much. Maybe a better example, and as beautifully described by Kessler in his, I think that's his name, in his book Up in Smoke. That's not his name, is it? Who wrote Up in Smoke? It was a Pulitzer Prize winning book about cigarette smoking, and he points out that prior to the 1870s, 1880s, that a different type of tobacco was really grown, and then the lowland areas of South Carolina and North Carolina became depleted, and they moved tobacco growing to these Piedmont areas. So it shifted, and they started growing a different type of tobacco called bright leaf tobacco. And bright leaf tobacco needed to be rolled into cigarettes, which started to be smoked around the time of the Civil War. And by around 1900, there was then a massive use of cigarettes. It was only at that time that physicians began to see lung cancer and all kinds of consequences of nicotine addiction, because it was a problem that really didn't exist in any similar form before the invention of the cigarette. So certainly the availability of new drugs, new ways to deliver it, and prices. I mean, as Marina pointed out about crack cocaine, that's when we really became aware of the addictive potential of crack cocaine. Apparently, using a little cocaine didn't hurt Freud very much. So there is this relationship then. But let's move to the genes. Let's talk about the functional alleles that are responsible for heritability. What is their abundance, and how do they work? Are these regulatory variants, or are they variants that alter the sequence of gene products, and what are the effect sizes of these? So this is a broad review, again, of genes and complex behavior, but some of these are directly relevant to addiction. The alleles vary from rare to common, all the way from observed in only one family, as in the MAOA stop codon in Brunner syndrome, to common, meaning that there are tens of millions of people in populations who carry the genotype. And the variants range from deterministic in their action, such that if you have the allele, that you will very, very likely have the behavior, too probabilistic in their action. So it just alters your risk by a factor of some magnitude. And they're both regulatory and also coding. And there are regulatory and coding variants that alter behavior at precisely the same genes. So for example, the serotonin transporter, there are a couple of coding variants that alter serotonin transporter function, and then there's the well-known HTTLPR regulatory variant at that gene. And here it is down here. This is the regulatory variant modulating anxiety. And similarly, here at MAOA, I'll be discussing, there's the rare stop codon that causes Brunner syndrome, and also there's the common short tandem repeat polymorphism, which just tends to make people more discontrolled in their behavior. So the answer to the question of if they're rare, are they common, are they regulatory, are they coding, is that they're of all types. And that means that different methods of discovery are needed to find them, and also that when they're going to be applied either in research or clinically, that they'll need to be applied in different ways based on the fact that they have different frequencies and different effect sizes. So here are two common variants that alter the risk of alcoholism, and in the Han Chinese population, and this is just a genotyping method that shows you the abundance of the three different genotype groups, homozygotes, heterozygotes here in the middle, and homozygotes here in red. So you see that these are common functional polymorphisms, both found in the Han Chinese population. And so how do those work then to alter risk? Well, here they are. They're altering the function of two enzymes that are involved in consecutive steps of alcohol metabolism. Alcohol is converted to acetaldehyde, which is a toxic intermediate that can produce flushing and upper GI cancer and discourage consumption of alcohol. And then the acetaldehyde is metabolized to acetate. And amazingly, in this population are millions of people who not only carry this or this, but who carry both. And so they are at different risk for problems. The ALDH variant was actually one of the genes that was genotyped as part of the Bring Your Genes to Cal program that met with some controversial headwinds a couple of years ago, because it was claimed that by the people who came up with this idea, which is a kind of a really nice idea because it's a good way to engage all the entering members of the freshman class into genetics and get them to learn something about genetics and feeling of community and all those good rah-rah things. But it wasn't done with any type of genetic counseling. It wasn't done with any view that there was significance of the trait, except that if you had the lysine 487 variant and drank a glass of alcohol, that it would make you flush. And so here is a flusher without alcohol, and then after a glass of alcohol. And in his photo, actually, it's permissioned, and this is from a PLOS One paper where it's all available online, so you can easily download these images if you should want. But it turns out that this is not an innocuous genotype. That first of all, if you're in a family where you might be interested in who's vulnerable to becoming alcoholic or not becoming an alcoholic, it exerts a 4 to 10-fold effect on odds of alcoholism. And second, if you're a moderate drinker, if you're an Asian flusher who has this genotype, you're at dramatically higher risk of esophageal cancer, because acetaldehyde is a pretty potent mutagen, and there are high levels of acetaldehyde. So it's this double-edged sword thing again. You're less likely to become an alcoholic, but if you are a moderate drinker, as many people are, nowadays. It's even thought to be, kind of cute or cool to drink alcohol and flush, then you are at higher risk of developing an esophageal cancer. There's also the fact that it's a paternity marker, but we won't go into that. Some of these incoming freshmen might have discovered that they had different parentage than they thought. Maybe they were adopted and didn't know it. There are things like that that you can actually learn from genotype. So I'm wondering, when should be the break for lunch?
It's up to you. Lunch is there, and what do you feel you'd like to break? You know you're talk better than I do.
I think it might be good to go on a little bit further now that I've mentioned this, because it'll heighten the anticipation and ultimate reward. So let's start with a little bit from heritability to genes. The main methods are locus-based linkage in families, which is often called genetic linkage, and allele-based linkage in case controls in families, which is often called genetic association. And in trumping all of this is really functional genomics, taking the genetic variants into other model systems and showing that they do what we think that they do, and tracing their effects within the humans through various methods, including measuring the intermediate phenotypes. So what is locus-based linkage in families? This was the old-time genetic linkage, which is now, once again, so important because of the recognition that rare alleles and uncommon alleles are of vital interest. So locus-based linkage in families can be performed in arranged crosses and model organisms, or we can use naturally existing families, going back to the original model put forward by Newton Morton, how to take just found families and get linkage information out of them. What one is doing is detecting meiotic linkage between nearby loci. The marker that is linked can be either coupled or in repulsion in different families. So let's say that there are two alleles, 1 and 2. Allele 1 can be tracking with the disease in family 1. Allele 2 can be tracking with the disease in family 2, because it's locus-based linkage, not allele-based linkage. It's just anything at the locus. It works well in instances where there is genetic heterogeneity within genes. So if you have 40 different mutations that can occur within a gene, in 40 families, each one of which has a different mutation, they will all add up to a very powerful genetic linkage score using family-based, locus-based linkage. It also worked well in the prior genomic era when we had small marker panels, because you could cover the whole genome with only a few hundred markers. And even today, it's overkill if you use more than a few thousand SNPs. That's all you need for locus-based linkage, and that's because, on average, there's about a 1 to 2% recombination rate per 1 million base pairs. So this is inherently a low-resolution method, no matter how many markers that you might genotype. And so people will sometimes say, well, we're going to refine our linkage map by adding more markers, but it doesn't really help that much if you're doing locus-based linkage. And for a whole genome linkage, the linkage structure of the human genome, this 1 to 2% recombination rate per 1 megabase, and we have 3,000 million bases of DNA. It means that a p-value of 10 to the minus third to 10 to the minus fourth is equivalent to a p-value of 0.05 after correction for false discovery. And sometimes that's not understood, even with family-based linkage. And it's masked a little bit by the use of log scores. So these p-values in family linkage are traditionally converted into log scores, which is just a log of base 10. So if it's 10 to the minus 3, that's a log LOD score of 3. But if you tell somebody that you have a finding that's, 10 to the minus 3, they think that's really good, but really it's just barely significant. So here's allele-based linkage. This is association and genome-wide association. It can be done in case control families or in, samples or in families. You can do association in families. It's done all the time. And there's some special methods that are used. One detects linkage disequilibrium to a functional locus. This means the non-random association of two things in the general population. It's like saying that if you have red hair, you're more likely to have the trait of hopping, or whatever random trait that you might imagine. These two traits would be observed to stick together in the population, or red hair and I don't know what else, short stature. Freckles. But the problem is that red hair and freckles are causally related, so you trapped me there. That's a causal relationship.
Pain.
Right. So this is allele-specific linkage because it's not locus-based linkage. We're asking whether a particular allele is related to the trait, whether we're doing this in a family or we're doing this in a population. And unlike the family linkage, it works very well to detect small allele effects because giant samples of individuals can be collected for whatever trait. And the genomic coverage is achieved with large marker panels on the order of 1 million markers. On average, the resolution is high. So the resolution is on average from 5 to 100 kilobases. For a single locus, the p-value threshold is, if you just did one candidate gene, is 0.05, just like it is for any other type of science, but there are a lot of caveats, which we're going to discuss. But for genome-wide association, the p-value that you need is approximately 10 to the minus 8 because of all the multiple testing. You did 1 million markers, which means you bought 1 million lottery tickets. And so that's equivalent to about 0.05. But as I say, there are a lot of nuances, not of all of which we'll be able to get into. You'll be relieved to know. So here are these markers and why it works. So on these different copies of the same chromosome, we see that there's a particular nucleotide that can have a substitution from C to T, and this other nucleotide can have a substitution of G to A. The combinations can then be put together in haplotypes, which are just like little words, like CAG, with the 3 different SNPs that vary here. And so you might imagine that if you have some local region of the genome that has a couple of hundred polymorphisms, that there then would be millions of haplotypes that would be observed in a population. And that's never seen. What is seen instead is that for any local region, any region of 40 kilobases or 100 kilobases, there will be a restricted number of haplotypes due to the linkage disequilibrium between these neighboring markers. And just to make a long story short, explaining all population genetics to you in 30 seconds, there's a phenomenon called genetic drift, whereby whereby generation by generation, there's a chance that any marker or combination of markers, just by chance, will not be transmitted to the next generation. If you have a population of finite size, it cannot support millions of combinations of markers. Instead, what will happen is that generation by generation, many of these combinations are lost, and then they have to be periodically regenerated by new recombinations, but the recombinations happen only rarely between markers that are this close on the chromosome. So you have a slow process of generating these new haplotypes, and you have a rapid process of getting rid of them by genetic drift. So, does genome-wide association work? Yes, it does. Here are the genome-wide associations that are posted currently on the NHGRI website for 17 different trait categories. And so what you see here are thousands of significant GWAS hits for these 17 traits, and many of them are overlapping. It's quite interesting that there are, the convergence of genome-wide associations for traits that should be caused by the same locus. And so it has worked spectacularly, but where are we at with GWAS in addictions genetics? These studies are detecting only a small percent of the variance in behavior. Furthermore, the individual loci are acting only very weakly. And this isn't just a problem in addictions genetics. You can look at the recent science paper on the genetics of intelligence, where a genome-wide association study was done on intelligence, actually it's educational attainment. And they found actually several loci that modulate educational attainment. If you have the marker, you're more likely to get a high educational attainment as compared to somebody who doesn't. But the effect on educational attainment is almost zilch. So for any one of us, if we had that marker, the effect would be almost non-existent. To detect the effects requires samples of hundreds of thousands of individuals who've been measured for educational attainment, and across the very large sample, even though the effect is significant, well, the effect size is still what it is, very, very small.
Height is another one.
I beg your pardon?
Height
Is another one.
Which is highly heritable.
Highly heritable, and the individual loci that have been found in GWAS are of modest effect. And meanwhile, via candidate gene analysis, there are, of course, very, very good height loci. I mean, everybody knows that the somatostatin receptor is involved in growth, because look what happens to people with a mutation in it. They are midgets. And similarly, everybody knows that that one mutation can lead to achondroplastic dwarfism in a very dramatic way. The person is half as tall as they would be if they do not have that variant. So you're always dealing, in genetics with these variants of very small effect, but that may be abundant in populations, versus one that are profound effects, and yet they're only important in that very small fraction of people who happen to carry them. So family linkage is detecting those loci that are undetectable by GWAS, and that's because of its ability to capture effects of rare alleles, and to some extent it may also be because people in the same families have undergone the same exposures quite often. They're at least from the same community. They are exposing them. They are undergoing similar exposures quite often. There are functional loci that have been found at several candidate genes that modulate addiction physiology and thereby risk, and the animal models are shedding light on heritability, but have found very few of the functional loci. So let's talk about context of effects. The genes are acting in context. It's not just that alleles have an effect size, but they have an effect size that depends upon the context and the phenotype that you're going to measure. These are a series of functional loci that modulate behavior and that are dependent on the context of stress. And the two most famous ones are the serotonin transporter and MAOA variants, which the Caspi and Moffitt group with the Dunedin longitudinal cohort were able to show had this interaction with either childhood trauma or trauma in young adulthood. And so here is the MAOA effect. This is the MAOA VNTR, and with low or no stress exposure, it doesn't matter if you have the low activity MAOA VNTR allele, but with severe stress exposure, then the low activity allele is associated with a high frequency of discontrolled behaviors, including suicide. And here's the serotonin transporter effect. There is no effect of it, or there's even a crossover effect at low levels of stress exposure, but at high levels, there is then this strong effect of this functional locus. So why should it be that there should be context dependence, and that studies which don't take into account context dependence sometimes can't find the effect at all? Well, one reason for that is that sometimes these effects are not just diluted, but sometimes they can actually be contravened if there are crossover effects. And we have an example of that with the COMT polymorphism. The low activity COMT allele, which is the methionine allele that I'm homozygous for, is associated with higher levels of dopamine levels in frontal cortex. And so one is already starting out on this inverted U-shaped curve of a relationship between frontal dopamine and cognitive performance. One is already starting out pretty much at a peak dopamine level from the standpoint of frontal cognitive performance. And so if you augment my frontal dopamine levels with amphetamines or stress, chances are I'm not going to do any better. So you can stress me all you want, and all it's going to do is probably lower my performance. I'm not going to, this is it. I mean, this is the best it's going to get. Whereas you can take somebody who has this valine 158 COMT genotype, the VAL, who has the high activity of COMT and is metabolizing dopamine rapidly and they have lower frontal dopamine levels, it is thought, augment their dopamine with amphetamine and stress. And then, here again with this, this ancient Pat Goldman-Rickich model of relationship of frontal dopamine to cognitive tuning in frontal cortex, get an increment in performance. And that was actually observed in this paper from the Danny Weinberger group, with first authors Venkata Matai. Now, conversely, these VAL genotype individuals who may not have quite the baseline cognitive advantage seem to be more resilient against stress. And there are many papers, including an imaging genetic study that we published a number of years ago in Science with Shankar Zubieta, showing that people with the VAL genotype are more emotionally resilient, and their amygdala and hippocampus respond less to emotional stresses than do people who have the cognitively more advantageous MET genotype. And so this is the so-called warrior versus warrior dichotomy for COMT. So you can then just see, if you're dealing with a population of people who've had very different stress exposures or maybe drug exposures, that you could be not only diluting your power to detect the allele effect, but you might not ever see it. And somebody else who has a different population might see a different effect of the polymorphism altogether. Now, I reviewed these G/E studies just briefly. Those of you who read the literature know that there are all kinds of controversies about these G/E studies. There are people who firmly believe that there's not one of these polymorphisms that is involved in a G/E interaction that has been replicably confirmed. And then there are others, such as myself, for other reasons, who think that several of these are quite solid. So what do we know from the meta-analyses? Well, the meta-analyses have told us that many of the gene by environment effects, such as described by Caspi and Moffat, are small or non-existent. So there is a proliferation of this literature. In addition to the ones that I put on that slide, there are hundreds of other G/E interactions that have been performed at this point. Many gene by gene effects would fall in the same category. And so it is important that people try to replicate these findings and look across studies meta-analytically. In meta-analyses themselves, the results may vary. So if you go to the serotonin transporter literature in detail and look at the different meta-analyses, it's quite startling to see that a meta-analysis will appear that proves that there is no G by E interaction of the serotonin transporter with stress, and then a month or two later, another meta-analysis will come out, also done by people who know how to do meta-analyses, that prove that there is that G by E. And how can that be? Well, the studies may include different studies in a meta-analysis. I mean, a problem with meta-analysis is that it can be a lowest common denominator thing. Does the study meet criteria? And in order to do that, if you want to get the maximum number of studies, then you use the very simplest phenotype available. Or you may exclude studies for one reason or another. And then there also is some difference in techniques. I believe also, though, that meta-analyses have the fundamental problem that the studies that are being added together are not necessarily created equal, and many of them aren't. The twin studies that we were considering earlier were ones that were really selected to be epidemiologic, and they were done by the best people. And it was in an era when there weren't that many people who could do studies of that nature, and partly that was because large twin studies are very expensive to do. You have to create this vast apparatus, and you have to be at the top of the field in order to do one of these large epidemiological twin studies. Not so for candidate gene association. Almost anybody can do a TACMAN assay for some genetic marker, collect a phenotype and a little bit of information about stress exposure, and publish a paper. And so the result of that has been a lot of bad papers. It isn't just that there's a problem with publication bias that favors the positive papers, but it's a problem that the literature contains a lot of papers that are based upon not very good science, not very good data sets, maybe nothing ever worked in the data set. And so then a meta-analysis doesn't inherently have a way of distinguishing between one study and another, except when the study was done, if some general methods were followed, and of course the size of the study. And the bigger the study, the bigger the impact on the meta-analytic result. I would say that the largest lesson from the meta-analyses is not to do lateral science. And so one's looking at an area where there's already good reason to believe that there's an interaction of a gene with some environmental parameter. You should make an assessment of that based upon the genetic linkage and association evidence, but also other information that may be available to you, including neurobiologic studies and including a careful review of the best studies that have been done, to establish the findings in the first place. Make up your own mind, and then do something else with the with the science. And what one really should be doing is seeking predictive validity via other models and at other levels. So I think I'll stop here because I don't want to delay reward too much.